Alright guys, today we're going to embark on a survey through biotechnology and some biotechnological techniques. We're not going to get real deep into a lot of these because they can be obviously relatively complicated in how they work and how they were discovered and things of that nature. But we're going to talk a little bit about how they work and a little bit about what they can be used for more than anything. Okay, so a lot of the stuff that we're going to look at here, has a little bit of content related to the viruses and bacteria, as you'll notice. It's not a lot, but it's enough, like I said, to put them at least in the same chapter. So, biotechnology. One of the first things I want to talk about with biotechnology is the use of something called a restriction enzyme. And one thing you'll notice about the biotechnology is that the use of restriction enzymes really permeate a lot of the uses of biotechnology. And what restriction enzymes are is that they are enzymes or proteins generally produced by bacteria that cut DNA molecules at certain DNA sequences, which are called restriction sites. Now, in bacteria, these enzymes are actually used in kind of self-defense. If you recall, there's a way for viruses to infect bacteria, and when they do so, the viruses put their DNA into the bacteria, and the bacteria doesn't really like that virus DNA in there, so they've got these enzymes to help chop it up. What human beings have done is they've kind of repurposed these enzymes to help us discover more about our genome and the genes that we possess as human beings. So the restriction enzymes generally make a bunch of different cuts, and they produce something called restriction fragments. And these restriction fragments, once they're cut, they can actually be reattached at some point because these restriction fragments have what are called sticky ends. And these sticky ends are complementary to other sticky ends, and when they find each other, and you'll see in the next slide, they find each other, they'll bond, okay? And then DNA ligase can actually seal up the little chunks of DNA as if they were never cut at all. So restriction enzymes are a DNA cutting tool, and humans have learned to manipulate this particular type of protein in an effort to learn more about our own genome. So this diagram here kind of outlines what restriction enzymes do. Let me get the pointer out here. There it is. Put me over here. So the restriction site, as you see right here, is highlighted in light green. And specifically, the enzyme that we're going to be using in this diagram cuts between G's and A's. Okay, so here we see here's a G and an A, and here's a G and an A, and the restriction enzyme is going to cut it at these particular points, and the DNA that was attached here is no longer attached, and it kind of breaks apart. So we've effectively cut it. And one of the things that we can do is we can take a little chunk of DNA and we can actually throw it in to these two cut areas and then reseal it. Because you'll notice that if we have a little chunk of DNA that has complementary bases to what's already there, it'll actually line up and match and kind of stick. And that's why they call it the sticky end. And so in this way we can actually manipulate DNA as well. We can take DNA that wasn't in our genome and we can put it into our genome, and that's what we see happening right here. And in the end we can use DNA ligase to seal the little gaps here and create basically one continuous strand of DNA. So there are multiple ways that we can use these restriction enzymes for our benefit. One of the first ones I'd like to talk about here is DNA cloning, and this DNA cloning actually kind of goes along with what's called bacterial transformation. And bacterial transformation is a form of DNA technology that basically allows us to produce certain genes, and not produce certain genes, but produce certain proteins based on putting the genes for those proteins into bacteria. So that's kind of what we're talking about here. And to put those genes into the bacteria, we have to use the process known as transformation that we learned the other day. Okay, so to work specifically with specific genes, scientists prepared gene-sized pieces of DNA in identical copies in a process called gene cloning. And to do this, what they do is they basically manipulate bacteria and their plasmids. Cloned genes are basically put right into the little plasmid of the bacteria, and then those plasmids are given to other bacteria, and then we know what bacteria like to do. They like to reproduce a lot. And so that's effectively kind of how that works, and we make tons and tons of copies of these things. So this is kind of what this is a real layman's look into how this works. You have a bacteria with the plasmid, okay, over here on this left side, and you have a gene of interest. We don't know what it is. Maybe it's a gene for laser eyes that we found in some animal. Who knows? I don't know. But it makes it more fun. So we find this cool gene of interest, and we want to make a lot of copies of this gene of interest because we want everybody in our army to have laser eyes. And so we take this gene of interest, and using restriction enzymes, we can cut it out of this DNA, and then we can attach it into an RNA, or pardon me, into a bacterial plasmid. Now this is just a nice little vessel. It's actually known as a vector. It's a nice little vessel to kind of house that gene until we can put it somewhere where we can make tons and tons of copies of it. So now we've got that little gene embedded into this kind of vector, and we put that vector into a new bacteria cell. And what do the bacteria cells do? Well, they reproduce a lot, which is why we see a bunch of copies of it here. And also, they go about their normal daily business, and they just produce protein. And so now there's also a protein harvested with it. So we can harvest a protein that's created by these genes, or we can harvest the genes themselves. Okay, so that's kind of the quickest look into DNA cloning. Okay, cloning a specific gene of interest. Now there are definitely I don't know why that's there. Okay. Yep. Okay. I must have accidentally copied something when I didn't mean to. But this is where I meant to go. So what we can do is clone eukaryotic genes in that bacterial plasmid and let it replicate. And so these are the 6 steps. And obviously there's much more that goes into this process here. But these 6 steps kind of go with these descriptions that I've got on these slides here. So one of the first things you're going to do is isolate plasmid DNA from the bacteria and also human DNA. Okay, we're going to cut both the DNA samples from the bacteria, okay, the plasmid, and the DNA with restriction enzymes. And the restriction enzyme is going to cut just around the gene of interest, and then all we need to do is basically combine the genes that we cut out here with these little cut plasmids, and they will, the little chunks from the human DNA will actually insert themselves into the plasmid DNA of the bacteria. And once we get these plasmid DNAs from the bacteria, complete with our little chunks of DNA in there, you see it's pink and black and yellow, we can then transform the bacteria, which means to have the bacteria take these foreign DNAs from the environment into their body, and then what we do is we plate them. Okay, plating the bacteria basically allows for us to select certain bacteria that we know have a specific set of genes in them, and when they are plated, they grow into these little colonies, and each one of these little dots here is billions and billions of bacteria, all with the exact same gene that we have embedded in the plasmid. So we can make billions of copies of some cool gene or some cool trait that we want to create, or maybe just some really practical gene that makes a practical protein that we can use to solve some kind of issue in the world. Okay, so it's an efficient and easy way of creating multiple copies of genes and also massive amounts of proteins that are produced by those genes, and we can do it with a very specific gene in mind, and we can do massive amounts of copies very, very quickly, because that's what bacteria do. They make copies of themselves really quickly. So how do we get that DNA in there? How do we get that plasmid in there? We can do something called electroporation. Electroporation is just a way to open up holes. You give the bacteria a quick shock, and it opens up holes in the cell membrane of the bacteria, and then the little plasmids will just seep right in there. And so the bacteria will be transformed. Or you can be a little bit more brute force and take like an actual little microscopic needle and inject your little plasmids into a specific cell, and then they can do what they need to do. Okay, so that DNA, that plasmid that we have, complete with the gene of interest for us, can be put into the cells in a couple of different ways. And when that DNA is in there, the natural genetic recombination happens where our genes just kind of take it up. Okay, it's just a natural thing that happens. Very similar to crossing over, where regular just recombination happens. If we put extra foreign DNA in there into our cells or the cell of interest of the animal that we're trying to manipulate or whatever, our DNA will just kind of naturally take it in, and that can be done in a couple of different ways, like we've just seen there. Okay. The second little bit of DNA technology that we need to talk about per the College Board is called PCR, polymerase chain reaction. Polymerase chain reaction can produce many copies of a specific target segment of DNA. And this is done in a 3-step cycle. It's actually, labs, if we had a proper machine, we could probably do it here. Those machines are a little bit expensive, but they're about the size of a computer, and they run, you put the stuff in there and they just go. Okay, and so these polymerase chain reactions, basically what they do is they take a very small amount of DNA and they reproduce it exponentially. It produces exponential growth of identical DNA molecules. Now that seems like something like, why would you want just massive amounts of one stretch of DNA? Because that's what it does, is it produces massive amounts of one stretch of DNA. Well, there are a couple of reasons. But first, let's talk about how it happens, okay? So it happens in basically 3 steps, as you saw before here: heating, cooling, and replication. If you look, there we go. If we look here, let's say we've got a specific stretch of DNA that we want to amplify. We want to make massive amounts of copies of it for whatever reason, which we'll talk about here in a second. One of the things we have to do is we have to unzip it first, okay? And the best way to unzip it, without being too delicate, is to use heat. Heat will denature the hydrogen bonds that hold the 2 strands of DNA together. It'll kind of rip them apart, like you see in that first picture here. And then we want to cool it. Okay, we're going to add DNA polymerase, and once we cool down this thing, the DNA polymerase and the primers can start their business. Okay? Cooling will allow for the primers to form hydrogen bonds with the ends of target sequences, and then we can include the DNA polymerase, which will extend those particular DNA strands until they're done. And so we just continue to do this over and over and over and over again. And ultimately, if we start with 1 strand even of DNA, we can just reproduce it exponentially. So 1 cycle of this process takes 1 strand and makes 2 new strands. And then we take those 2 new strands and we make 4 new strands. And then we take those 4 and we make 8. Okay? So it is a process that takes small amounts of DNA and amplifies them exponentially. And that's important because we're going to combine this type of DNA technology with one we're going to cover in a second. But with some other DNA technologies, you need large amounts of DNA. Certain things that scientists use are not able to detect 1 strand, but they're able to detect 5 billion strands. So if you can make a ton of DNA, it makes the jobs of some of the other scientists and DNA technologies that much easier. Okay, so polymerase chain reaction is something that, as I mentioned before, can be utilized in many different ways. One way is crimes. Okay, so we see that's a drop of blood with a little crime, like a little label on a crime scene, and it can take small amounts of DNA found at crime scenes through various avenues, whether it be blood or sperm or hair or nails or whatever, skin cells, stuff like that, small amounts of DNA, and then it can amplify them greatly. And when you amplify that amount of DNA that was very, very small to begin with, then you can start running identification tests on the DNA that you actually have. Okay, and that's the important part. It can be used to date certain things as well. The one that's most pertinent to us right now is with COVID, right? And you'll notice this. I took this thing over here in the box. This is from our school's exclusion policy, and I've highlighted a part that maybe you've heard of or seen, and maybe just like, well, okay, I need a specific type of test if I want to come back to school. I need a negative RT-PCR COVID-19 test. And so what this tells us is that they ran this test using polymerase chain reaction, or at least that's what it tells me. Because if you have a very small amount of the virus in your system, it might not be enough of the DNA that's there to actually detect it when you use the test initially. But if you amplify the amount of DNA that is in the sample using polymerase chain reaction, all of a sudden it is detectable through the test that we actually use. So it's a way to get a more concrete evidence that you've been infected with COVID or some other disease. And it doesn't just work with that. They use it to diagnose all sorts of diseases because, if you are, infected with something, right off the bat and you kind of feel something's wrong, you might not have a whole lot of virus in your system at that point. It probably has not replicated to the point where there's a lot of it, and their genomes are so small. You want to be able to replicate their genomes a lot so that you can actually test to see if it's there, that the test that you use actually can detect the amount of DNA that's there. So they use polymerase chain reaction to make more of that DNA that was in your body at one time. Okay. All right, section 2 gets into restriction fragment analysis, and this is again where I talked about the importance of restriction enzymes, the fact that you have these little enzymes that cut DNA at very specific sites. Restriction fragment analysis detects differences in the nucleotide sequences of DNA molecules. It provides comparative information about DNA sequences, oftentimes can be used in DNA fingerprinting. One of the terms that you'll hear a lot when you talk about this type of DNA technology is called RFLPs, restriction fragment length polymorphisms. It's one of the more fun kind of things to say, RFLP. What this means is that the restriction enzymes are used to create little chunks of DNA. And these little chunks of DNA are all relatively, they're different sizes, but depending on how much DNA you have, you might have a lot of these little chunks of different sizes, okay? So it is a method for detecting minor differences in DNA structure between individuals, and these differences in the individuals, these patterns, are actually inheritable. And so if I were to take my DNA and administer a certain set of restriction enzymes to my DNA, and take Joe's DNA over here and administer the same exact restriction enzymes to his DNA, the restriction enzymes would cut his DNA different from the way it would cut mine, because I've got a different sequence of A's, T's, C's, and G's than Joe does. The restriction enzymes are going to cut my DNA at different spots, thus producing chunks of my DNA that are different sizes from Joe's. Okay? And so now, how do we determine what the heck that means? We're talking about all these different DNA chunks. Well, we use something called gel electrophoresis, and this is something you've probably seen. You've at least seen these pictures in those crime dramas and stuff like that, right? Well, the gels that we see here, this is like literal gel. It's like a hard gelatin, like hard jello. What it does is it allows us to separate all of these chunks that are produced by the RFLPs, by the restriction enzymes. So let's say, for instance, I've got this test tube here which has my DNA in it. It's been cut by a certain set of restriction enzymes, okay? And then we'll say the second vial has Joe's DNA. which has been cut with the exact same restriction enzymes. And it's got a bunch of different chunks of DNA in it, the same as my tube has a bunch of chunks of DNA in there. Okay, so the way that we do this is we put all of these chunks. These chunks have varying sizes. There's big ones, there's small ones, there's medium-sized ones, there's chunks that are all sizes in between. Okay, and so all of those chunks of DNA are in that fluid there, and we can dump that fluid into these little chambers in the gel. And so what we do from there is we would run an electrical current through this gel. We'd send electrical current going this way, okay? And so what ends up happening is that DNA is actually negatively charged, okay? And the DNA sits in these little compartments up here, these wells. And when we start to run the electrical charge through the DNA, what happens is this negative charge is coming here actually repels this DNA, and it actually works its way through this gel. Now these gels they're porous. They have holes in them, okay? So the little chunks of DNA are going to work their way through these gels, and you're going to see chunks of DNA that are migrating from the negative end to the positive end because DNA is negative and we've got the negative electrode down at this end. So depending on the size, those little chunks of DNA are going to work through this gel at a different rate. So think about somebody who's very, very small. They're going to be able to wiggle into some of the smallest places that maybe a larger person wouldn't be able to get through. That's the exact same thing that's happening with DNA. And what we notice here is that the DNA that's shorter actually travels a further distance during a given amount of time because it can squeeze through those little holes in the pores of the gel more efficiently than the larger chunks, which kind of get stuck up at the top. They're so big they can't work their way through those pores as well as the little chunks. And so what we have here is we have a way of identifying that these are the large chunks, these are kind of the medium-sized chunks, and these are the smaller chunks. And each person, aside from identical twins, are going to have a different set of chunk patterns. That's kind of what I would call these, chunk patterns, from somebody else. So here would be my chunk pattern, right? And here would be Joe's chunk pattern. And the reason we can look at this and identify different individuals from one another is because our DNA was administered the exact same restriction enzymes, and it cut our DNA at different points, yielding different size DNA chunks. And so those different DNA size chunks are going to go through that gel at a different rate, and it's going to cause them to line up differently. So I can unequivocally say that this person here who produced these DNA chunks is a different person from this person here who produced these DNA chunks. Okay? Now this is oftentimes used in many different things. One of the first things we'll talk about here are identifying two different types of alleles. Okay, so you can identify two different types of alleles for a given gene, and also we use these in paternity and murder tests. And I'm sure there's countless other applications for this particular technology. But gel electrophoresis can analyze two different alleles because depending on what allele you have, you will probably have a different set of A's, T's, C's, and G's. So when you're looking at an allele like, for instance, this one, allele 1 has a specific DNA sequence, CCGG here and then CCGG here, and a restriction enzyme is going to cut between the C's and the G's and basically make a certain amount of chunks. But allele 2 is going to have a different DNA sequence. Instead of CCGG, it's ACGG. Okay, that's what makes it allele number 2. That's what makes it a different version of that same gene. But what we notice here is that allele number 2 just doesn't have the second cut. Okay, there's not a second cutting site for the restriction enzyme that's used. So you're going to get different numbers of chunks based on the alleles you have. So if you have allele 1, let's say it's sickle cell, maybe you produce 3 chunks, W, X, and Y, when your DNA is cut with the restriction enzyme. If you have allele number 2, maybe it's normal, you only have 2 chunks, Z and Y. And so in a gel electrophoresis, when you ran this DNA through the gel, you would see 3 chunks would indicate you have allele number 1, or 2 chunks you have allele number 2, based on how the restriction enzymes cut a specific allele versus another. Okay, and so we can see that here as well. Sickle cell gene, I had them flipped around, but regardless, not a big deal. So that's how we see the pattern shape out with sickle cell. And then DNA fingerprinting, we can kind of see this. This will be one of the activities you kind of have, is trying to solve a crime with using DNA fingerprinting. You've got all sorts of different samples here. You've got the victim's blood. In this case, this must be a murder or assault charge or something like that. You have blood from the defendant's clothes, so the person they think is the one who did this. They took some blood samples from their clothes. So right off the bat, what this tells us is that the victim's blood, okay, produces this chunk pattern, is the same as the chunk patterns that were found on the defendant's clothes. Okay, so something's fishy there. That means the victim would have had to have been close enough to the defendant for that person to get, for the victim to get their blood on their clothes. So it's used as a piece of evidence to associate the defendant or the perpetrator with the victim, and the fact that the victim's blood was on their stuff. And we notice over here the defendant's blood produces this chunk pattern, which is totally different from the victim's. And we notice that because, you know, lawyers are kind of tricky sometimes, right? They might come out and say, oh, well, that my defendant, the client just had a bloody nose. That's the blood that was on there. And so they'll try to talk that away. And then, so this is a piece of evidence trying to refute that part of the statement, saying, well, this is the defendant's blood. The pattern that's on the defendant's clothes does not match his blood. It actually matches the victim's blood. Okay, so it can be used in evidence for that as well. So it's a kind of a fascinating use for that. The third bit of DNA technology that we're going to talk about is different technologies that have been used to map the entire genome, or sequencing DNA. We're basically going to boil DNA sequencing, whole genome sequencing, the Human Genome Project. We're gonna boil that down into two slides, which is next to impossible, but we're going to try, because you don't really need to know a ton of detail about it, and the detail, frankly, is tricky. There's a whole lot that goes on to it, so we kind of have to give you the big picture of what they did to sequence the human genome. But yes, the human genome has been sequenced. This is kind of a map of all of the a lot of the pertinent alleles, but genes that can be found on human chromosomes, and provide some information about what you would see in terms of gene mapping. And so the Human Genome Project spent lots of time trying to figure this out and put it together so we have a better understanding of exactly where our genes are located and what they do and how they might interact with other genes in our actual genome. So it can be the researchers of the Human Genome Project sequenced the genome in 3 stages, and they used multiple techniques. One of the first things that they can do with our chromosomes, specifically our condensed chromosomes, is that they can fluorescently label genes of interest. Okay, that's a whole other topic in and of itself, so we're not going to really get into it, but know that they can fluorescently label these genes, and so you can figure out where these genes are located on the different chromosomes when they light up. And so what that does is it gives the Human Genome Project basically a target. And it says, okay, we're going to look here. We know there's a gene, let's say right here. The little yellow band indicates there's a gene here. We want to know all about it. We want to know the sequence of it. We want to know the A's, T's, C's, and G's that make it up. We want to know all about this thing. That's the ultimate goal, is to get a letter-for-letter understanding of each gene in our body. And so the first thing that they would have done is taken and created a genetic linkage map, okay, which is similar to what we would have done. Remember we did with the fruit flies, kind of made a linkage map, same type of thing. They look for recombinant frequencies, and they look for these genetic markers. And these genetic markers can be anything. They can be specific sequences that have been labeled. They can be specific genes themselves. Regardless, they're able to lay this out using genetic markers. Okay. The next thing they want to do is physically map these things. And what they do is they use, again, restriction enzymes to chop up stretches of DNA. So they take maybe this stretch of DNA right here, this little stretch right here, which actually is probably comprised of, a couple thousand base pairs. And they use restriction enzymes to chop it up into a bunch of little fragments, like you see here in this diagram with the mouse, or with the laser pointer. Okay. So it chops those up, and then what it does is it takes these fragments that we see here and it runs them through in a computer. And it effectively lines them up and looks for overlapping segments. You could theoretically do this as a human being. It would get awful tedious and boring after a while. So they've trained computers to do this. You chop up these fragments, and then they see, okay, this fragment right here has some base pairs in common with this fragment here. That must mean that they overlapped at some point, and then they can deduce what is the sequence later on. And so then they use a sequence called, I got to look it up, dideoxy chain termination method, to actually sequence what the letters are in one of these specific areas here. Okay, and dideoxy chain termination method uses radioactively labeled base pairs, and we're not going to get into it. I'll see if I can find a video that kind of explains it, but it's not going to be on the AP test, like an in-depth understanding of this. But effectively what it uses is fluorescently radioactively labeled base pairs instead of the normal base pairs. And it takes a specific DNA stretch and actually grows it, and it can recognize, the computer sequence can recognize which fluorescently labeled base pair is there, and then it can look at them in order and deduce what is the actual letters, or what are the actual letters that are found in a specific stretch of DNA. It's really quite ingenious when you think about how they do it. I don't know how they think these things up. You got, like I said, for science, it's as much technical as it is artistic, and this is one of those really, I think, elegant artistic ways of deducing what the DNA sequence of a given stretch of DNA actually is. So again, I'll try to find an animation that kind of illustrates this and put it in with some of the information you're going to read here today, but or listen here to today. But I didn't want to overburden you with a little bit extra stuff that's not going to be on the test. So I hope you enjoyed that. It was quite a quick foray into DNA technologies, and I sped through a lot of it, but at least you now have a little bit of understanding of some of the major important things that people utilize to make new discoveries in science. And we'll talk to you soon.